Thanks for the nice introduction, and thanks for having us. So we will be presenting about our recent work which we published in Nature Genetics about the identification of rare and common regulatory variants in induced pluripotent stem cells using population-scale transcriptomics. So I'll be presenting the first part. I'm Marc, and Craig will take over roughly halfway. And just let's get started. I guess you all know, but just to get everybody on the same page, we've been highly successful in linking all kinds of different traits to different sites over the genome. So we actually have now links to over 2,000 different traits, over 500,000 different unique sites on the genome. So here's an outdated screenshot of the NHGRI EBI GWAS catalog. And each of these dots represent a group of different traits linked to a specific site on the genome. And just to give an example, for instance, for height, we have over 900 unique variants that all give a tiny increase or decrease for changing your height. But how these variants, so the variant here on the left, how they all work together in giving you a slight in or decrease for developing disease or changing your height, that we don't really know. So this is still sort of a black box. So for some sites it's easy, for some it's more difficult. But one of the ways to look into this is using quantitative traits loci mapping, or in our case specifically EQTL mapping. So to do this, we basically compare genotypes to expression of a gene. So here we have the third gene with the telomerase reverse transcriptase gene, and this red line here represents a variant. And what we see is if you stratify the expression of this gene in iPSCs based on the genotype, you see that when you have the minor allele, the TT allele, you have high expression, and when you have two copies of the more common allele, you will have low expression. So this is an example of an EQTL. Of course, there's many more variants in this locus. So here you see a Manhattan plot of the gene region, and you see here this red line. In this case, our top variant for top EQTL variant in iPSC is actually also linked to cancer, and there's multiple different other variants here, the blue dots, which are linked to cancer. But we also see that this is a very different regulatory pattern from what we see here on average in the bottom over all of the different GTEx tissues. So the iPSC regulatory landscape is very different, and it can be interesting for cancer like we see here, but also for development. So to study this in a larger context, we started the RTQTL Consortium. And here we're looking at we're trying to define the genetic regulatory network in iPSCs. We focus on structural variation, rare variants, something that Craig will talk about. And we also try to look at distal acting variants. And all of this together hopefully helps us to get more insight into the molecular mechanism of disease via EQTLs and other genetic association studies. We won't really talk about the structural variant part. Craig will mention it a bit, I think, but this has been published before. So if you want to look it up, it's been published in June of last year. And here's the two titles. Within the consortium, we reprocessed and reanalyzed data from 5 different cohorts: the HIPSCI cohorts, the IPSCOR cohorts, genes IPS, Philips, and IPS samples from the 1,000 genome study. And in total, we analyzed close to 950 unique donors, and we had a total of 1,400 IPS lines we looked at. So for some donors, we had more than one IPS line available. For all of these samples, we had chip genotypes and RNA-seq data. And for some of the lines, so 425 of them, we also had whole genome sequencing data. So that's where we could look at structural variation and also look at rare variants. The first thing we did is basically comparing our expression patterns to the expression patterns within the GWAS consortium. So you can see our data here on the top right. We see that it nicely overlaps with ESC data, which we also reprocessed, and we see that our fibroblast lines look very similar to GTEx data, although Of course, there's some batch effects, and there's also differences between the different studies, but this is not a major thing when you look at the first two MDS components. We find close to 1,800 unique e-genes when you look into our sample. When you compare this to GTEx here on the left, or a large EQTL study on over 2,000 unique blood donors, you see that we have relatively high power. But it's actually even close to the 2,000 EQTL map on 2,000 samples. And we also see that here is in the other marked boxes, there's the studies, the original IPS studies, we see that we have 2.5 times more power to identify EQTLs as compared to what we had in the initial studies. So this gives some insight into how different these regulatory networks are as compared to GTEx. But we also looked into tissue specificity. So this we did using Measure. So here you can see in red the sharing of EQTLs based on the effect sign in GTEx. So you can see that over all the different tissues they studied, on average, they share 90% of their EQTL signals. And I mean, there's some which are much lower. Similar from the other GTEx tissues. But our iPSC data actually looks quite different from the GTEx patterns. So you can really see that there's a difference in the genetic regulatory landscape in this early time point as compared to what you see in GTEx. And to look into this a bit more, we combined our data with a recent differentiation study by Como et al. And you can really see that when the iPSCs when we differentiate the iPSCs towards endoderm cells, that the regulatory pattern starts looking more and more like GTEx. So here, again, we have violin plots for our own study, and here's the IPS stage, mesoendo stage, and endoderm stage of the GTEx, of the Como study. And when you compare this to the expected signals from GTEx, you see that the more differentiated you get, the more similar you get to the somatic tissue genetic map. So we also looked at trans associations. So here will be an overview of the trans EQTLs we find. So here we have the genic loci here on the y-axis and a SNP locus on the x-axis. And we find over 193 unique independent trans EQTLs. There's a lot of variants which are linked to different EQTLs, over 800, but we only find 191 unique genes. We could replicate our findings using DNA methylation data and left out expression data. For this, we actually specifically looked at our cis-EQTL variants, and we looked at GWAS variants. 21 of the trans EQTLs were only linked to GWAS, so actually quite a bit less than what we see for EQTLs. And we also see that the trans EQTL map is quite different from what we saw in other tissues, so there's very minimal sharing. Again, we looked into this Como et al. Study, and we see that the further you go in differentiation, the less of the trans EQTLs you can still replicate in our single cell data. Very quickly, we also looked at using our data for interpreting GWAS signals. So here you can see a genetic loci on chromosome 1, close to the DPYD gene, and this loci is also linked to waist-hip ratio. You can actually see that we see quite some sharing here between the landscape, basically, of the GWAS hit and the EQTL signal. So we specifically tested this using colocalization, and you see quite strong colocalization between the genetic variant, the EQTL variant, and the genetic variant for waist-hip ratio. We did this basically for all the publicly available full GWAS summing statistics, and this is what you get. You see strong colocalization for height and waist-hip ratio, but of course these are also traits where there's many loci that are linked. But we also find, for instance, for multiple sclerosis or coronary artery disease, so we can actually interpret more loci than we can using the GTEx data alone. And when you put this in numbers, we see that there's 836 of these loci, which we can only interpret using iPSC and not using any of the 48 GTEx tissues. And this is a direct overlapping set. So we specifically looked at the same studies and the same data, basically. All this data is available in Locus Compare. So if you want to look up your own specific variant of interest or site of interest, you can look it up there. And I will hand over here to Craig.
Okay, thanks, Marc. Okay, I think everyone can see this now. Let me know if there's any issues. So just briefly, I want to present this slide. So the slides I'm about to present, what it relies on is a expression outlier approach. So this is an approach enabled by RNA sequencing, which allows us to measure individuals' transcriptome, or the expression levels of genes across the genome. And outliers here are individuals who have markedly less expression. So the person on the left here, an underexpression outlier, or more expression, or an overexpression outlier, the person on the right, and compared to a large reference cohort, here the RTQTL iPSC transcriptomic information. And this approach allows us to assess the functional consequences of coding and non-coding variants. So the plot on the left from GTEx shows that there's an increased likelihood of observing rare variants within the genes of individuals who are outliers across a range of transcriptomic phenotypes. And here, there's 3 levels shown: gene expression, splicing, and allele-specific expression, or ASE. And the trend here was quantified as relative risk, which is shown on the y-axis. And it increases as a function of the frequency of variants. So rare variants show larger enrichments. And what this is suggesting is that rare variants found in outlier individuals could be driving large effects on transcriptomic phenotypes at multiple levels. And so we're interested in running the same analysis in a comparative analysis between RTQTL iPSCs and GTEx. And the plot on the right shows what we found is the strongest enrichment is obtained in iPSCs when compared with somatic tissues in GTEx. So with this observation, one thing we wanted to look at in the paper was modeling the molecular consequences of pathogenic rare disease variants in iPSCs, and particularly in known disease genes which are not expressed in blood. And so in these plots, we're classifying genes into 2 buckets of expression on the x-axis, with this first bucket containing genes which have really insufficient expression for identifying outliers robustly. And on the y-axis is the percentage of known disease genes from OMIM, the Online Mendelian Inheritance in Man database. So OMIM genes from various disease groupings, which fall into each bucket. And so we found that across all disease categories, more genes are expressed in iPSC than blood. So within the RTQTL cohort are individuals with rare diseases who were enrolled in the HIPSCI study, which is one of the studies incorporated into the RTQTL Consortium. So now using the RTQTL reference dataset, we're able to model expression outlier events in iPSC in known causal disease genes for these 2 distinct rare diseases. And these histograms here show the gene expression z-score of rare disease cases highlighted in red compared to the full RTQTL cohort in gray. And for both these diseases here, we see clear outlier expression patterns. And looking at the 1 patient case in more detail, that of hereditary cerebellar ataxia, if we look at the distribution of gene expression across all tissues in GTEx, we see that the causal disease gene is mostly only expressed in the brain. And here are those tissues in more detail. But now, as illustrated in the dashed black line box. If we add in the RTQTL iPSCs now, we can see we see expression now in iPSCs of a gene that has clear tissue-specific expression patterns, and particularly in tissues that would be very dangerous to biopsy clinically. Now, briefly, just switching gears to look at rare variants in common diseases and traits. So in the plot on the left is what we're seeing is the absolute effect size, GWAS effect sizes of these outlier linked variants. So these are rare variants which we identify in expression outliers in I2QTL. And those are shown in the blue lines. And to generate a comparison set, we also looked at rare variants found within individuals that fall right in the middle or around the mean of the distribution of expression in these genes where we find these outlier individuals. And that just gives us a reference set to which to compare with these outlier rare variants. And then we stratified using the colocalization results. We stratified by colocalization clip score, which is just a measure of confidence in colocalization. And what we can see from that is there's an increasingly large difference in the absolute GWAS effect sizes of outlier and these non-outlier or matched control variants as a function of colocalization clip score. So taking a step back, in genes where we know that differences in expression of that gene are linked to significant effects on a trait or disease, we see that the variants linked to gene expression outliers tend to have larger GWAS effects than variants in our matched control or non-outlier variants. So this gives us confidence that integrating outlier expression information here helps to define a set of rare variants with large effects on expression and on downstream traits and diseases. And just to give an example of that from the paper, there's a SNP on the right highlighted with its RSID. And this SNP doesn't pass genome-wide significance, so it's nominally significant. So you would usually discard the SNP just using a p-value threshold approach. But the effect sizes in the top 1% of all SNPs in the entire GWAS, and I think in that GWAS there was about 10.5 million SNPs measured, and it's in a gene that's highly colocalized in the common variant colocalization analysis, and this gene is known to have a role in type 2 diabetes. So here, integrating outlier expression information gives us just an extra piece of information in order to interpret this GWAS result. And in the paper, we give a couple of other examples where the expression outlier information was helpful. So just to wrap up, because I think we're right on time, we discussed that integrating multiple data sets, iPSC data sets, gives us power to map both cis and trans EQTLs in iPSC. One thing we mentioned in the paper is that we actually find close to 1,000 cis genes that were not previously reported in EQTL maps from somatic tissues. We also show through the master analysis and some other analyses in the paper that iPSC genetic regulation is very different from somatic tissues. We also briefly highlighted that trans EQTLs may arise from GWAS and other EQTL molecular phenotypes that are in addition to just overall mean gene level. And then we also discussed that EQTLs linked to disease variants gives more insights into disease mechanisms. And finally, that the molecular consequences of rare variants can be modeled in iPSCs for both rare disease diagnosis and interpreting complex trait biology. So as mentioned at the start, this was a collaboration between several labs, mostly Oliver Stiegel's lab at the MBL and DKFZ, and also the Montgomery lab at Stanford. But we also worked closely with Kelly Fraser at UC San Diego and members of her lab, and also with Casey Brown at the University of Pennsylvania. And I think with that, I think there's a chance for questions.
I have a question, if that's okay. Can you hear me?
Yep,
go for it.
Yeah. So the donors for the iPS cells, do they come from people with disease specifically, or are these healthy individuals?
So mostly, the vast majority are healthy or apparently healthy individuals with no known diseases, but there is a subset, about 80 samples in HIPSCI are from known rare disease individuals. So about maybe like 7 to 8% of the entire cohort.
I'm curious, for the rare disease patients, did you have access to their parents' whole genomes and RNA-seqs as well?
No, we didn't, no.
Because that would be really, I've always wondered about parents of patients with like recessive rare diseases, and if they would fall in, if they were carriers, if they would fall in outlier levels as well. Something I've always been curious about.
That would be very interesting.
Sorry, for follow-ups or sort of a side study together with some other people here at the EMBL, we did actually have parents. Actually, we had fathers only, and then it's a it's a male-only disease. I don't remember what the disease was exactly, but there we could really see that only the children fall out. But you can very nicely use the fathers in this case as control as well.
Interesting.
As well as the rest of the RTQTL Consortium.
Interesting.
I have a question. Yeah, it was very interesting talk. So is that particular gene expressed in an iPS cells? Because I'm wondering if certain genes are not expressed in iPS cell, then that will be tricky to measure the EQ. It would be impossible to measure EQTL and trans-EQTL.
Yeah, so of course we're always limited to the expression level. Sorry, Craig. Of course, always limited to the expression level. I mean, for the example that Craig showed, you could really see that the highest expression is in blood, and it's relatively high expressed in iPS. But it's not. Of course, it's not. We can't do anything when there's just not enough expression. The only thing which we do see is that there's relatively many genes expressed in iPSCs, so many more than any other easily biopsyable tissue like blood or, for instance, skin. But it is much in that sense. It's more interesting to also look at iPSCs for other diseases that are not necessarily visible in blood, for instance.
Okay, thank you. So then, just follow a quick question. How many, like it's a rare variant, as you mentioned, how many for your target have you validated that they are really following your pattern and hypothesis?
So we're very limited right now because only a subset of a very small subset of the rare disease samples in HIPSCI have whole genome sequencing also available. So we'd like to look at both variants that fall within the coding and non-coding regions around candidate rare disease genes. But right now we just don't have the data in order to do that.
Thank you.
I have one final question. Oh, I was just going to ask what your thoughts are on integrating these outlier variants in like a polygenic risk score to, I guess, combine the results from the EQTLs and these outlier variants to predict patients.
That's actually something we have started to think about now. We didn't work on that much for this paper, but we have several ideas for follow-up papers, and that is definitely one of them.
Cool.
I have a question related to previous questions. So in your specific example, that rare disease tissue of origin you mentioned was like hard to get. So it was a brain sample, a brain disease. And in that case,
yeah,
why do you think
sorry,
the iPSC expression pattern capture it particularly well compared, because you thought that the stem cell is pretty far from a brain cell. Yeah, so I'm just wondering what's your thought?
Well, I think like overall, you just in effect like turn on the expression of many more genes, and I don't think it really matters which tissue they're eventually expressed in a somatic state. So I think it's just especially for that example where the disease site is the cerebellum, which would be I don't think you would ever really get a clinical biopsy of that site. But the fact that it's why is it expressed in iPSC, I think we're just in the process of generating iPSCs. You're just turning on the regulatory programs of many more genes, and you're able to just see the expression of those. It's not universal. You can't capture every gene, but certainly like a lot more. I don't think it's biased towards any particular lineage like brain tissues or other tissue sites.